Let's go to the 17th questions. Now, question number 17. Which of the following statements is correct? Animals can show mitotic divisions in both haploid and diploid cells. After a space, the number of chromosomes becomes double. 2 and 2, 4n. Chromosomes not going to double. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. SR synthesis phase marks the period during which RNA synthesis takes place. Is mitotic division both the haploid and diploid cells? No. Option C here is right, because during the G2 phase, proteins are synthesized to get cell division occur properly. So, question number 18. In which of the following stages a chromosome is minimum coiled? That is interphase, because remaining phases are included in M phase, which is most condensed stage. So, option A here right. 19th question. Which of the following statements is correct regarding G0 phase? Mitogens are present in G0 phase. Mitogens are present, but energy rich components are absent. Both mitogen and energy rich components. So, D, neither mitogens nor energy rich compounds are present. S, D. In G0 phase, there is no mitogens and no energy rich compounds. Here option D is correct. Now question number 20. Best material for studying mitosis in laboratory is leaf tip, shoot tip, gamete. C, option C, root tip. Leaf tip and shoot tip are not correct. Gamete is also not correct. Option C. Question number 21. Mitotic spindle is mainly composed of dash proteins. Tubulin, myosin, actin, D, ectomyosin. Tubulin. Option A. 22 question. Microtubules are absent in mitochondria, flagella, spindle fibers, centriole. Here, mitochondria is absence of microtubules, because flagella consist of microtubules. 23 question. Centrosome undergo duplication during dash and begin to move towards opposite poles of the cell during dash of dash stage. Centrosome duplication occurs in S phase of interphase. Begin to move towards opposite poles because in the anaphase in case of mitosis. So option B is suitable. Now question number 24. Dash is the best stage to count the number and study of morphology of chromosomes. In case of study of chromosomes or observation of chromosome number, anything or behavior, our metaphase is perfect. So option B is correct. As we can see, 24th question. Dash is characterized by the all chromosomes coming to lie at equator with one chromatid connected to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fibers from the opposite pole. Okay, equator. So here option B, metaphase, is correct. All chromosomes lie at equator in the metaphase. Now question number 26, read the following statement. In mitotic cell division, chromosome number is hollow. Centrosome is a point where two sister chromatids are held together. The period between two successive mitotic divisions is known as telophase. In G1 phase of cell cycle, protein RNA are synthesized. Now we write is like this. So here we have to find out the correct options. So option first one is wrong. Mitotic cell division, chromosome number is no, remains same. Centromere is point where two sister chromosomes are held together. Yes, it is right. The period between successive mitotic division is known as telophase. No. G1 phase cycle, protein and RNA synthesis. Yes, it is right. So here 1 and 4 are right. So option C. Now question number 27. You are provided with floral buds of chrysanthemum in your class and are asked to count the chromosomes. Then which of the following stages would you prefer to look into? Count the chromosome, metaphase. Option B, 28 question. During karyokinesis, the spindle fibers get attached to condensing chromosome at a highly differentiated region. This region is called as spindle fibers attached at kinetochores. Option D. Now, as you can see, if this assumed as centromere, this is chromosomes. These tiny structure is called as kinetochore, where spindle fibers is get attached. So one chromosome, two spindle fibers attached to each kinetochore. Now question number 29. You have to identify the markings. Second one, central, no, centriole, yeah, second centriole, fourth chromosome, first chromatid, third centromere. I think option A is correct. Option A. You can see now. Let me erase this. Now question number 30. Which of the following statement is not correct regarding colchicine? It prevents assembly of microtubules. It inhibits chromosome replication. It is an alkaloid. It is called as mitotic poison. So here we have to find out the wrong one. Colchicine can do. It prevents assembly of microtubules. It is right. It is an alkaloid. It is right. It is called an mitotic poison. Yes, it is right. So option B is wrong. It inhibits chromosome replication. No, it is wrong because chromosome replication occurs in S phase. Now the colchicine works at metaphase. So there is so much difference. Now 31 question. Colchicine is a cell poison which arrests cell division at dash and can induce dash. It can arrest at metaphase and induce polyploidy, where chromosome number increases. So as you can see, colchicine where it acts. So there will be no spindle assembly and no splitting of centromere. So the 4C amount of DNA remains at that, and it becomes chromosome number becomes double. So that's why polyploidy happens. It occurs in metaphase. So option C. 30 question. Spindle formation can be disrupted by exposing cell to the microtubule pathogens such as colchicine. Option D. 33 question. In at which stage of mitosis the two daughter chromatids separate from each other, migrate towards the opposite poles, and are now referred to as chromosomes of the future daughter. So this is anaphase. Option C. So here the chromatids are separated and goes into the poles. So anaphase is right answer. Let me erase it. Now question number 34. During anaphasic movements of chromosomes of each chromosome is or the pole and dash of the chromosome trails behind. Arms trails behind centromere. Option E. As you can see, assume it as a cell and here is the chromosome. So here centromere is pulled out and the arms are trails behind. This occurs in anaphase. By this, chromosomes are given a name to their shape while in anaphase. So as you can see, metacentric it is. As you can, let me explain in a practical way. Let me take any property to explain this. As you can see, metacentric chromosome show V shape, submetacentric show L, and icocentric show J, and telocentric show I shape. So assume it as a chromosome where I touch this here, you can assume it as centromere. Now this is arms, assume it as arms. Now as you can go up, you will see like a V shape. Now as you go towards the end, as you can see here, it is little bit of L. Now as it goes to the end, as you can see, you will find it as a J shape. As you can close to this, you can find it as I shape or like one equal bundle, one bundle, just like that. I hope you understand. Let's get into this question. Now question number 35. Select the correct match. Question phase, G2 phase, no. Synthesis phase, J1 phase, no. Centromere splitting, anaphase, right. Chromosomal condensation, telophase, no. Chromosomal condensation occurs in prophase. So option C, centromere splitting in anaphase is right answer. Now 36th question. The separation of two chromatids of each chromosome at early anaphase is initiated by. Here, the interaction of centromere with the chromosome fibers, the elongation of metaphase spindle, the force of repulsion between the divided canicrackneetifistartnow, all of this. So here the answer will be the force of repulsion between the divided canicrackneetifis is right answer, because in anaphase the chromosomes move towards the poles by the contraction of spindle fibers. So at the time of anaphase, the splentromere is splitting happens. So that's why you can see in anaphase, at a late phase of anaphase, you will find a chromosomes with two arms or a chromosome with one chromatid. So 36 answer will be C. Now question number 37. Match column 1 with column 2 and select the correct matching. So you can match this one. V shape is for metacentric, L shape for submetacentric, J shape for acrocentric, and I shape for telocentric. Here you can observe the options. You will find it easily. So option B is right. 38. Which of the following is key event of an? As you can see in 38 question, chromosomes move to spindle equator, get aligned through spindle fibers to both poles. Wrong. Centromere split and chromatids separate. S. Chromosome cluster at opposite spindle poles and their identity is lost at discrete elements. Here discrete elements means it symbolizes the prophase. So it is not recording the anaphase. So here option D, both B and C, no, it's also wrong. So here option B will be correct answer for this. In case of option A, it is referred to the metaphase. Now we can move to the next question. Question number 39. Select the incorrect match regarding the matrix cell division. Prophase chromosomes begin to uncoil. Metaphase chromatomes move apart. Let's move apart now. Telophase non-nuclear memory appears. Okay, late anaphase. Each chromosome consists of two chromatids. Each chromosome consists of two chromosomes. Late anaphase. No, interphase chromosomes are not distinct. Yes. So we have to find out the incorrect answers row. So here 2 and 4 are incorrect. So option A will be right answer. As you can see, assume it is a chromosomes. Now centromere is split. One chromatid goes up and one chromatid goes down. Option A is correct. Now, as you can see, what is true about telophase stage of mitosis? Chromosomes lose their identity as discrete elements. Chromosome cluster at opposite spindle poles, nuclear unpole, nucleus Golgi, and plexus reform. So here for telophase, option D is correct. All of them are true. So we have to go to the next question. So you can see this 41 to 44 questions. We have to label them the diagrams first. Then we can answer the questions very easily. As you can see, so first question, 41 question is like identify in the diagrams. It is like anaphase. Okay. Now B is like metaphase. Now C is like prophase. Now D is like telophase. So anaphase, metaphase, prophase, telophase. Option C will be right. You can check them. So option C. Now 42 question. Which of the following shows the correct sequence of the given matrix table? So by the labeling you can understand which stage is first and which stage is last. By this we can arrange them. First is C, then after B, after that A, and after D. C, B, A, D. This option D is right. And now get into the further question. At which of the given stages of matrices chromosomes appear in VLG? Okay, anaphase. Anaphase belongs to A. So option A is right. Now we go on to the next question. 44. Read the given statement which represent the features of the figures A, B, C, D and match them correctly and select the correct option. First one, chromosomes appear like a ball of fuel. Reformation of nuclear embryos. Here, 2 is for telophase. Formation of equatorial plate. This is for metaphase. Splitting of centromere. This is for anaphase. Chromosomes appear like a ball of fuel. This is going to be prophase. By this we can arrange easily. As you can see, here prophase is 1, telophase is 3, 2, and metaphase for 3 and anaphase for 4. So by this 4, 3, 1, 2. You can check the answer. For 44 question, the answer will be A. Now let me erase this. Now question number 45. Match column 1 with column 2. Select the correct option from the given quotes. Disintegration of nuclear membrane. Prophase. Appearance of nucleolus. Appearance. Telophase. Division of centromere. Anaphase. Replication of DNA. S phase. A2, D4, D3, C1. Option A is right. Now question number 46. Which phase of mitosis is essentially the reverse of prophase in terms of nuclear changes? So in case of prophase, what will happen? The reverse should be, we have to think. So it is reverse to the telophase. So option C. Now question number 47. The given graph shows the change in DNA content during various phases. Today, metatis identify the phases and select the set one. Okay, now ABC for G1, S and G2 and M. So option C is correct. Now question number 48. A cell's division time is 1 minute in 20 minutes. A culture tube is 1/8 filled with cells when the tube will be fully filled. So here we can solve this question as no, the cell division time is 1 minute. Now it is 1/8 filled. Okay. Now for each cell division it becomes double. So by this we can solve this question as like this: 1/8 into 2. This is first division. After that, after the first division, it remains 1 by 4. After the second division, it will become half cup full, and after the third, it becomes 1 and it will be completed. For this, we took 3 divisions. First, it is 20 minutes. After that, 3 minutes is adding. Now, total time will be 23 minutes. So, option B is correct. Now, question number 49. Cytogenesis in an animal cell takes place by dash method in dash direction, while in plant cell it occurs in dash method in dash direction. In animal cell it is furrowing method. In case of plants it is a cell plate method. Furrowing is centripetal and cell plate method is centrifugal. So here furrowing is outwards to inwards. In case of cell plate it is inwards to outwards. So option B is right. Now question number 50. If a tissue has at a given time 10-24 cells, how many cycles of mitosis had the original parental single cell undergone? From one cell, these cells are formed. For becoming 10-24 cells, how many divisions it happened? It is gone through. So we have to find them. So it would be like 10. As you can see, 1 to 2, 2 to 4, 4 to 8. We can increase the number like this. We get 10 divisions for 10-24 cells from one cell. So here the option B is right. Now 51 question. Spindle usually persists in form of dash during dash method of cytogenesis. Okay, fragmoplast as cell plate. Fragmoplast and cell plate, it occurs in plant cells. So option B. Now 52 question. Matching the column. Division of nucleus, karyokinesis, division of cytokinesis, DNA replication occurs in S phase or interphase. We can say karyokinesis is not followed by cytokinesis. Syncytium. Multinucleotides is happened. It is called as syncytium. For 52 question. Division of nucleus A4 D3. A4 D3. Option B is right answer. Here for 52 question, option B is right. Now 53 question. The given diagram depicts the cell plate method of cytokinesis in plant cells. Identify A, B, C. Here A is that is daughter nucleus and B will become cycloplast and C is vesicles. So A is the daughter nucleus and B for cycloplast and C for vesicles. So here by the options, option A is right. Now let me erase this. Now 54 question. Phlogistoblast is related to division of nucleus, cell elongation, cytokinesis, assembly of chromosomes, cytometaphase. Option C is correct. Cytokinesis. Now 55 question. The role of mitosis is not merely to divide a cell into two daughter cells, but to ensure genetic continuity from one cell generation to another cell generation. The mechanism ensuring genetic continuity is. So option B and C are confusing because formation of two daughter cells. Okay, it is right. Formation of two cells with identical name, it is also right. But in case of option C, it is more accurate than B. So here option C is right. Now question number 56. Chromosomes duplication without nuclear division refers to. Chromosome duplication without nuclear division refers to. Here it is called as endomitosis. Now question number 57. Meiosis consists of two cell divisions without any DNA replication. No. Two cell division in which chromosome number is reduced to half. Yes. Two cells division with only two rows of chromosome replication. No. A single division with chromosome replication. No. Option B is right. Two cell divisions in which chromosome number is reduced to half. First meiosis 1 division. Meiosis 1 is like reduction division, real reduction, and meiosis 2 is like equational division. Now question number 58. Which of the following is not the future of meiosis? Meiosis involves two sequential cycles of nucleotide cell division. Meiosis 1, meiosis 2, but only single cycle of DNA replication occurs. Yes, okay. Meiosis 1 is instead of the parental chromosome have replicated to produce the identical sister chromatin at the phase. Meiosis 1 is instead of the parental chromosomes have replicated to produce identical sister. No. Meiosis involves pairing of non-homologous. Okay, B is right. Non-homologous chromosomes and recombination between them. Here, homologous chromosomes appears. So option C is wrong. Now question number 58. So option C will be right answer because it is wrong. Meiosis involves pairing of homologous chromosomes, not non-homologous. Now here 59 question. Best material to study meiosis is. So here we can observe options: root tip, ovary, pollen grain. Here root tip is vegetative part, not correct for this one. Ovary and pollen grain are developed not in condition of meiosis. So young anther will be answer. So option C is correct. Now question number 60. The term meiosis was given by. This is informative. Option D. Farmer and Murray. Now question number 1. At which of the following stages the chromosomes appear single, thin and thread-like? No, this is will be leptotin. Starting phase of prophase 1. Leptotin. Option A. Now zygotism of prophase 1 is characterized by formation of cytotonical complex. Option D. Now 63 question. Crossing over in diploid organisms is responsible for dominance of the increase between genes, segregation of alleles, recombination of alleles. Recombination of alleles. Option D. 64 question. Which of the following is correct in the given figure? Okay. As you can see, in case of 64th question, in diploid organisms, the meiosis occurs in the reproductive system for recombination of alleles. As you can see, 64 question. My remaining videos are not like this. This one is very slow, but remaining videos will be very fast. Which of the following is correct regarding the given cell nucleus? Number of homologous chromosomes here 3. Number of chromatids will be okay. Number of centromeres. So here option B is correct. Now question number 65. The number of homologous pair of chromosomes. Yes. The homologous chromosomes are identical in size and appearance, but not in having same genetic materials. They don't have same genetic material, but appearance and size are similar. So option A will be right. The remaining options are not correct. Now 66 question, which of the following is correctly associated pair of homologous chromosomes at the start of meiosis? Okay, here as you can see, we go through the options first. You can see option A here. These are not in identical size and appearance. So one is large and another is very small. So this is not correct. In case of option B, these are like mirror images, not in correct accurate size, and the chromosomes difference is at each chromatid, but it should not like this. So option B is right, not correct. Okay, in case of option C, here sizes and appearance is similar, but it is also similar like option B. So here, as you can see, option D, it is more correct. So option D is correct. Now 67 question, which of the following is correct? About bivalent. Bivalents are tetrads, yes. Bivalent means 4 chromatids and 2 centromeres, correct. One bivalent consists of 2 homologous chromosomes, okay. Bivalents form in zygote in yes. All of them are correct. Option A is right. Now 68 question. The process of crossing over is assisted by which of the following enzymes? So it is particularly recombinase, but here we have to select them by the options. We will select okay A and C, endonuclease and ligase, because in case of crossing over, the chromosome material is exchanged by cutting off the one of the arms of the chromosomes by other arm, in which where it happens between the homologous chromosomes of different genetic material chromosomal material. So option D is right. Now 69 question. To build up food reserves in the cytoplasm, chromosomes become unfolded to start generation of mRNA and RNA during which phase of meiosis? It occurs in diplotin. As you can see, diplotin. After the diplotin, diakinesis is appear. After the diakinesis, prophase 1 is completed and metaphase 1 is started. So for this 69 question, option C is correct. Option C. Now we go into the question 70. From this question, part 2 ends. Don't forget to watch the part 3.